Hello and welcome to a new season and a new episode of the Computomics podcast. We have a great guest today to start us with this season. She's the group leader of automated plant phenotyping at Leibniz Institute of Plant Genetics and Crop Plant Research in Gatersleben, Germany. Welcome, Kerstin Neumann.
Yes, thank you, Anna. I'm happy to be here.
We're glad to have you. As you know, we start with a little icebreaker here at Computomics podcast. And a little birdie told me that you might be into gardening. So I was wondering, what's your favorite part about growing vegetables in your own garden?
I think that's pretty much the experience that you are responsible to have a plant growing from seeding up to harvest, whenever that is. Yeah, can be different for vegetables, fruits, etc. And that it's basically your creativity or your care that, leads to the success or a failure. I mean, depending, right? So there can be weather conditions where the failure is not your fault, but it's kind of this little bit godlike gardening, which I really enjoy because you decide this is growing here, this is growing there, and what you do with the plants and what you are using them for. But on the other hand, it's for me totally relaxing also to take care of the plants after a stressful day, to water the plants, to go a little bit taking care of the plants. That's really totally relaxing for me.
Oh, that sounds like a dream to come home. And you can also even use some of your work knowledge, obviously, in gardening, I'm sure. Let's switch to that, actually. As I introduced you, you work at the Leibniz Institute of Plant Genetics and Crop Plant Research in Gatersleben, IPK for short. What is your primary focus of research there?
So I have to explain maybe a little bit better. So the group that I'm leading is a infrastructure/research group. So we are managing very diverse phenotyping infrastructure at IPK that is including a worldwide unique facility, the Phenosphere, where under strictly controlled condition, you can enable field-like growth and non-invasively follow that via camera systems and sensors and really do very complex experiments. And you design the scenario under which the plants should grow, even under future climatic conditions, for example, under increased CO2 concentration, that is also possible. So it's technically very challenging. And then we have several automated plant phenotyping systems where the plants are grown in pots, and these pots are standing in carriers, and they are moved automatically through the imaging towers, and you have also automated watering. So there is also a kind of technical team in my group that monitors that the systems are running, that takes care of further developments, and also if something is not working, then trying to repair or restart, etc. So there's a lot of technical infrastructure we take care of. And then on the research side, of course, we are using these facilities to explore non-invasively the growth of plants. And me personally, I'm doing that for cereals and more recently also for legumes. And we have this focus of abiotic stress tolerance. So we have dedicated drought setups, and also we are currently working on drought and heat combination, for example, as a stress factor.
This specific research you just mentioned, you're using the Phenosphere for, is that correct?
Yes, we are using the Phenosphere, but we are also using the already established automated plant phenotyping systems. So both projects cover both of it. Yes.
Can you go into a little more detail? Just take us through maybe the project setup for the cereal stress research. I'm just going to paraphrase it.
Yes. So we started basically years ago with a plant genetic resource panel. That means it's not modern varieties. That was barley, by the way. And this was a panel developed from gene bank accessions of IPK. And then we tested the drought stress setup because it's also complicated to work with that system. So you have to do a lot of testing before. And then we were sure that we have a good setup. We were mimicking basically a spring drought event. So you have in the tillering phase, so before flowering of barley, you have already the tiller formation where later the spikes develop. And in that phase, we were having a 3-week drought period, which you can nicely simulate with this automated watering station. You have full control over the watering or not watering, and also over the weight. So you can follow the weight means the water loss for each individual plant on the system. And then we also looked on the recovery. So we had a rewatering event and then basically rewatered back to the normal irrigation level, and were able then to see the recovery. And see means that the system doing images with cameras. For example, RGB cameras from top and side already give you a hell lot of information. There is a so-called biomass proxy, which you can get from the plant area of the side view and the top view camera, which we call biovolume. And that corresponds very nicely when you destructively would measure the fresh weight or the dry weight of the shoot. I mean, you only see the shoot, of course, in the image. In that system, we don't see the roots. And then you can, by that proxy, when you do daily imaging, you can follow biomass development over time in the two treatments, means in the well water treatment and in the spring drought treatment with recovery. And we used that information in the barley collection. We also had genetic information. That means they were fingerprinted. We had SNPs available, and then you can do a statistics that's called GWAS, genome-wide association mapping. So you basically have a statistic model that uses the genetic information and the phenotypic information, and you find out which genomic regions are important for that trait. And as we have that trait over time, we use that GWAS with the biomass data of every day. And that's actually very interesting, because then you really can track the involved genomic regions, where should be candidate genes regulating that trait, across the observation period, which was for us from seedling stage up to, let's say, flowering stage. And you see that there are different genomic regions involved between early and late growth. Now, leaving out the spring drought, so that's already very differently regulated, different genes, different genomic regions are involved here. And you can really see that the so-called significance levels are appearing and fast disappearing, or are slowly increasing and then staying stable for the, let's say, later observation period. You have more stable genomic region. There's not so much change anymore. So that's already very complex studies. And then you add the drought on top. So everything changes. You find some genomic regions which are shared between the irrigated and the drought stress treatment. However, there is a shift in importance of that genomic regions. And you find also then different genomic regions which are only appearing under drought and not at all across the timeline in the well water treatment. And then again, you have the shift to the recovery. And you see also a switch back in the genetic architecture, meaning, again, different genomic regions are important for that recovery period. And this is with one experiment, you can now really track non-invasively the growth of plants and the involved genomic regions, candidate genes. Of course, we followed up on that. So we went deeper. We had different collaboration partners where at important steps in the drought, we also took samples. So we sampled leaf tissue that goes for metabolomics. So you want to look on what's inside the plants, what's happening. And then also in the recovery, this is still under, let's say, investigation. So we don't have the final results yet, but it looks promising to also get further insights there. So that was the first step. And we applied that from barley, going also to elite wheat panel, where we saw a similar pattern and also, similar behavior of the genetic architecture of biomass over time. We added with a new camera at that time, because it's really, I do that since quite a long time. I had now I call it now here a photosynthesis camera. It's a little bit more complex than that, but basically you can follow the photosynthetic efficiency of photosystem 2. And that changes, of course, due to the stress. So you have a decrease of that photosynthetic efficiency under drought stress, and you have a very fast recovery when you rewater the plants and back to the normal level. So they are not permanently damaged, at least not with the setup that we were using. And for example, we saw in wheat, where we had this data, and then we followed the plants until maturity, means we harvested the grains at the end of the experiment and did a lot of grain yield components, that the photosynthesis in the recovery period was also connected with grain yield under stress. Not at all in well-watered condition, because photosynthesis is not a limiting factor for grain yield when you have normal irrigation. But our assumption is that when you have the rewatering period after stress, that the plants try to catch up. So they try to form the biomass that they couldn't do under the stress period, and then they grow a lot. So you have a higher relative growth rate compared to control conditions.
Mm-hmm.
And then photosynthesis can be a limiting factor. And that's why the plants that can do that a little bit better in that period end up with higher grain yield in our setup and in our greenhouse.
Mm-hmm. And have you compared different breeds, if you will, in their performance, or is this firstly about really understanding kind of the system through these different phases?
No, of course. I mean, the collections that we were using were different varieties. In case of barley, it were older varieties coming from the gene bank. In case of the elite elite winter wheat panel, I should say, it was a breeding progress panel. That was a Brivex project financed by the BMBF. And that were 220 winter wheat varieties from, I think the earliest were from the 1960s, and then up to the start of the project, which was about 2012, I think the newest varieties in that time. And what we see is a clear effect on the year of release. So it were always the top yielding cultivars from that times. And when you compare then the phenotypic data with the year of release, you see, first of all, a reduction in biomass, a reduction in plant height, but of course an increase in grain yield. And what we also saw is an improvement of photosynthetic efficiency, for example, which can then compensate it for the reduced biomass and account for the improved grain yield.
That's amazing. I'm guessing this non-invasive method really allows you to see that full system at work too across different varieties. Over 50 years and 220 varieties, that's quite a sizable amount of data too.
Yeah, it is. I mean, you have the imaging data. Of course, you have 3 side views, 1 top view. You have different camera systems. So it's a large data set. If you go for 60 days or longer of phenotyping, we have the luck that we have an in-house image analysis group here in IPK, and that connection works very well. So they take care of analyzing this data, because you can't work with the images. Statistically, you need to get out traits in form of CSV file, for example. So they have to first separate plant from background, and then, of course, give you traits out of your plant height, plant width, the area. There are traits regarding the color values from RGB, for example. So what we, for example, can also follow in stress is the color value, and also more specific ratios of color, like the yellow to green ratio, or also the red to green ratio. And you have an increase in the yellow to green ratio under drought stress, which is due to the fact that some leaves are wilting and dying off. That's also a stress indicator then in the data set.
And what role does AI play in this analysis, given that there is so much data?
Yes, a huge role, because the whole image analysis is based on deep learning methods. So they are constantly training the algorithms based on ground truth data, depending, of course, because you need to train. Ground truth data means you have a set of images which you annotate manually. So that's done in the image analysis group. And then you have a perfect separation of plant and background. And then you train the algorithms to match that data set very nicely. And when this works very nicely, you can apply this algorithm to the whole data set. And whenever you change parameters, you have to start again training ground truth data. That's a little bit annoying, but it gives you really, really nice and robust results. So, for example, when you work with wheat or barley, which are cereals, and you suddenly have legumes on the system, which we did, then you have to train the pipeline again.
Speaking of legumes, you are also a part of a very big European project through the Horizon 2020 line called Increase. Can you tell us what is this project about?
Yes, the INCREASE project is, first of all, as you rightly said, a huge EU project that is already going now in the 5th year. So we started in Corona times in May 2020, and it has 26 partners from 13 different countries. So it's a large consortium. And it's all about the utilization of plant genetic resources of legumes. So there are 4 legume species included in the project, which is the most outstanding of the project, I would say, most visible, but we come to that in a minute, is the common bean. But we also work with lentil and with chickpea and with lupin. Mm-hmm. And all the different partners. So first of all, we establish material. Now we come a little bit to the gene bank topic. So plant genetic resources are basically the lost or the diversity that is in the risk of being lost due to the breeding that is only using a few varieties. All the old varieties before, or even the farmer selections, they are lost to most of us, and they are conserved in gene banks. And we want to use that gene bank material, but before you can use it, you need to go through single seed descent. Single seed descent if you want to work with them genetically. Because on the genetic level, a gene bank accession might be homogeneous, might contain just one genotype, but it might also contain a mixture. And then you cannot really work with them genetically. So you take a single seed from that gene bank accession, you single seed descend it, you have from that plant then seeds, you fingerprint, and you use that single seed descent line for your experiments. So the INCREASE project builds up such material for the 4 legume species we were talking about, fingerprints them. So it's a lot of genomics in the project, including pangenomics, so really sequencing different genomes of several such lines. But also having field experiments, so agronomic field trials for a defined subset of a diverse collection, which captures most of the diversity. So you have to imagine we come from the full single seed descent panels, which are thousands of lines from different gene banks. Then you define a core set that captures most of the diversity to be representative for the whole panel. Mm-hmm. There you have deeper genomic information, and then you also add phenotypic information in terms of field trials, for example. Here at IPK, again, switching back to the automated plant phenotyping, we used, for example, the chickpea diverse panel that has denser genomic information on the high throughput systems, again, for early drought tolerance, because that's something they're not really adapted to. So they are very, very tolerant for terminal drought stress where they come from, but it's not really known how they would perform here in our environment where we have a lot of spring droughts. Mm-hmm. And this is what my PhD student did. And we are now working further with that panel. And we have a Humboldt candidate here, so having a Humboldt grant that right now has a second experiment on the system and testing combined heat and drought stress. So this is basically what we did in INCREASE regarding high throughput phenotyping. But the most outstanding part of the INCREASE project is for sure the citizen science experiment with common bean. And that's one task of the work package that I'm leading. And together with the coordinator of the project, Roberto Papa, I am leading this citizen science experiment.
And what is the goal of the citizen science project with the bean?
Oh, we have not only one goal.
Tell us all of them.
I mean, coming maybe the closest to what we discussed before is that we want to evaluate a large panel. So these are more than 1,100 single seed descended plant genetic resource lines coming from the INCREASE pool that we have, which are already almost 10,000 lines, and want to generate phenotypic data for this large collection. That's very ambitious. It's a very large panel, and we want to have that across all European growing areas, which are very diverse in terms of temperatures, climate, of course, also the day length, so photoperiod. And we thought the best to reach that is to employ the citizens. And of course, then not every citizen tests all 1,100 lines in their gardens. That would be a little too much. They get a subset of 5 old lines and 1 modern line. And the modern line is only included for statistical integration of all the data. So this modern line, everybody gets the same, and then everybody gets a different subset of these 5 plant genetic resource lines. And with that, and with the help of an app, so data are collected using the Increase CSA app, mostly in form of pictures, but of course in form of dates like flowering date or maturity date. We collect a lot of data in a very sophisticated way, and have over the years now established a large database for this panel. And we already see that the data look really promising. Of course, you have to work with a lot of missing data when you work with citizen science data, right? It's all voluntary. Everything can happen in gardening. Plants can also die. There can be diseases. There can be heat waves, like we had in the past years, especially in southern Europe. Or this year, it was very wet in our region. So a lot of people had slug problems, like me too. So they were once completely eaten by the slugs, and then 5 out of 6 lines, in my case, recovered. So everything can happen on the way. Plus, you miss the time. Now you can change jobs, you can change apartments, whatever. So you have a lot of missing data. But over the years, we really have a nice database now. This is only one goal, to generate phenotypic data. We achieved that goal. And then, of course, we want to use that data also for the genome-wide association studies. So they're all fingerprinted, and to explore the genomic regions which are important for the traits, and the traits that are important for the adaptation of the common bean to all the different environments. That's mainly the main scientific goal. We have 2 other very important goals, which are not so much scientific. The second one, and we also reached that, is to bring this topic of plant genetic resources, of legumes, why legumes are good for our agrobiodiversity, for our health, to the general public. Yeah, you can reach that nicely via the Citizen Science Project. Of course, we give out information. There's a lot of information on the website. We have Facebook groups where we can also inform the people on their topics. Especially of agrobiodiversity. I mean, we're in newspapers, there are TV reports about us, and this is all helping us to achieve that goal. And then we not only want that people participate once, we want to establish a community that has a decentralized approach of conserving these old varieties outside the gene bank. And conserving means also using. So by using these varieties, growing them, harvesting the beans, keeping some seeds for the next year, you preserve also these old varieties. I call it now old varieties. It's also linked races, etc. So non-released varieties. And this is also now achieved, I would say. So this year we had more than 1,400 people that came from the previous rounds that participated again with their own harvest. So we give out seeds only once, for example. When you participate the first time, you get the seeds from us. We decide what bean lines you get. If you participate again, you can decide, okay, I grow these varieties again, or only a subset of them, or I use the app and get in contact with other citizen scientists that continue and can request.
So you can swap seeds.
Yes, you can not only swap. So, for example, if you didn't harvest anything and you cannot offer seeds, you still can request seeds from others in the community.
Got you.
Or, as you said, you can swap. You can offer your seeds where you have enough to share with others, and also test simply other varieties. And this is nicely going, I would say. So there was a lot of activity at the beginning of this year. And also in the Facebook groups, people connect, and they met for seed exchange parties, for example. And that was really nice to see. And the long-term goal is really to have this long-term. And you know that funding is not long-term. So the Increase project
usually isn't.
would end at a certain point next year. But we had really worked hard over the summer. There was a new EU call on minor crops. And we submitted on the 17th of September a new proposal to continue with that approach. And in a different call, so there's also more in this project than, again, just a citizen science experiment. but we would like to continue with that and also extend it a little bit for more exotic bean species.
Mm-hmm. Interesting. I mean, obviously, we don't yet know if that call is going to be greenlit.
No.
Have our fingers crossed that it is. But I think you showed really clearly that you already have quite sizable results from this project. It's running since 2020, you said, right? Through 2025?
Yes, so the first round with the citizens was conducted 2021. So we started mid of 2020 with the project, and regarding the citizen science part, we first needed to develop the app. Means we needed to think about what we want to have in the app and etc. Develop all the phenotyping schemes, and then you need to gain participants. So the with the first growing season in the project, which was 2021, the citizen science part started.
Mm-hmm. And did you see an increase in participation? Because the way you describe it now, it sounds like the fire caught on in a way. But I wonder what that means in numbers, like how many thousand people participated in the beginning versus now, for example.
Yeah, so that's we were already very successful in the first round in terms of total numbers. So we had more than 3,400 participants in the pilot round, and we were also very surprised by that. Basically you run through open doors. If you say you work on old varieties and people can get seeds for free, they really love to test exotic material, which is not available in the shops. And also the topic of agrobiodiversity, I think, is in most of the countries already in the minds of the people, and they are really willing to participate. So we were quite successful in terms of total numbers in the first round, but we didn't have a nice distribution across Europe. So I think 2/3 of the participants were from Italy, because
2/3.
Yes, because Roberto is very nicely connected and he made it into national TV. And that's super difficult here in Germany with gardening topics, especially outside the gardening season, because the recruitment we do over winter to then have the numbers for the new round in spring. So that's really, we struggle with that here. Then we had, I think, in the first year, Portugal was number 2, and then number 3 was Germany. And then the others there were already many countries participating, but with low participation numbers. So from that pilot round, we learned a lot, and we further improved the app. We also knew how we should do the dissemination. And one important point, of course, is that you don't do the dissemination in English. If you really want to go to the regular citizens, that's a very important point and a very time-consuming point. You need to address them in their native languages. And that's what we did from the start. So we created like a voluntary translation team in the consortium, and then covered most of the languages present in Europe, or at least most of the main languages. There are some smaller countries where we don't have partners, like the Czech Republic, for example. We don't have a translation here. But Italy, France, Spanish, Portuguese, even Hungarian now, due to a voluntary citizens that was helping us later.
Oh, nice.
German, of course, is covered. Polish is covered. Slovenian. I'm forgetting some. So I think we are now at 12 languages altogether. So all the app is available in all these languages. The website content regarding the citizen science part is available in the languages, and all the dissemination is done in the local languages. So, and you can't do that alone. So it's a little bit more complex, right? So I can't do the dissemination all across Europe. I'm not speaking all these languages. I'm not having the local contacts. So we employ the whole consortium. So everybody is involved in the dissemination. We provide dissemination material, and they can also translate it into their native language, and then they use their local networks. So, and then what we did from round 2 on, to have a more uniform distribution of Europe, we gave weekly updates on the registration numbers to the consortium by email. And then you could see where there were gaps, and then people were also motivated. It was a little bit like a country contest, right? And also in round 2, Italy won, but very close behind was Spain, and again, Germany third in the second round. And I think this were more than 4,000 participants. And then came round 3. And round 3 was last year. And basically, we were knowing too well what we need to do. So we also had support from non-partners, so whether it were stakeholders of the project or organizations that we got in contact with, for example, the Meise Botanical Garden in Belgium in 2023 was doing a great just dissemination in Belgium, where we had few participants the years before, and suddenly we had more than 1,000 participants in Belgium. And also we knew better how to address the media here. So suddenly we had over 2,000 participants from Germany last year. So we had in total more than 9,000 participants in the round 3. And now comes the but. The but.
Okay. What is the but?
The but is that you have to distribute the seats to the citizens. So it's not you press a button and they all get their seats. They have to be packed. The right 5 varieties for everybody have to be packed into.
Even who gets what is probably a decision you have to make, right?
It's a huge effort, and you cannot thank enough the people of around the team of Roberto in Ancona. He employed so many students over weeks to pack the seats. And we were challenged again, like the years before. It's not when you use the regular mail, you really encounter problems across Europe. So it's really slow. We first thought it's due to the pandemic, because we had started in the pandemic situation, right? So the first round was 2021. So all the delays in sending, like it takes like 2-3 weeks to send seeds from Italy to other countries, but it didn't improve. So again, 2023, it took very long. And of course, with all this way more numbers than we expected, they also needed to pack a lot of more bags, and you could not send out all the seeds at the same time. So some people received it too late, and it was really months of work, for the Italian team. So in the end, we managed. Some seeds came very late, and then they used it in the next round, for example. But we said we need a kind of maximum number, and this is 5,000. And this year, we reached that number almost. So it were 4,900 something new participants, plus the 1,400 continuing participants that we didn't have to give again seats. So this was running well, and we so far could always accept everybody that registered to the citizen science experiment. But it's a kind of logistic challenge, so it's not that easy.
Yeah, it's challenging on many levels, and I think you've really taken us on the journey with with you and your team of like 26, you said, project partners. I think we will definitely have a link in the show notes so people can check out the project. Is there anything you'd like to maybe invite people to check out from your end?
Yeah, I mean, we will have still round 5, that's for sure. That's still running under increase. So registration would start typically around mid or end of November. And that information is available on the website, the pulsesincrease.eu. And then you have to go to experiment, and then it's also in all the languages. And then you can read how you can register, because you need to do that via the app. So you definitely need to download the Increase CSA app. Create an account there, verify the account, and then you have to inside the app register for round 5. And then you could be part of the Bean Friend community next year.
The Bean Friend community, that sounds amazing. I'm sure a lot of people listening to this podcast might have gotten the urge or know someone who's into gardening who might be interested. So we'll make sure to link to it in the show notes. You've described so many challenges, but also rewarding aspects of the project. What is maybe one that is especially prominent in your mind at the moment?
Yeah, that's very easy. So the most rewarding moment of the whole project was, of course, that we won this year the grand prize for citizen science of the EU. And we were recently on the 5th of September at the Ars Electronica in Linz. I haven't been there before. It's a great experience, very nice. And the whole team was there, and we received the award. And that's, of course, very nice for the project. So we get some money that we can also use for the project. And we want to use it to improve the app. And also we haven't talked about it. We have a lot of schools on board. So a lot of school gardens across Europe also participate meanwhile. And for example, we would like to develop some children-friendly background information material for the schools.
That sounds amazing. So all you listeners out there, you've heard it. You can participate, or if you know someone who's into gardening, if you're related to a school that has a garden, check out the show notes, check out the project. You might be able to participate in a pretty amazing citizen science project. And thank you, Kerstin, for your time in taking us into that project.
Thank you very much, Anna.
Thank you for listening in today. Want to learn more? Then check out our show notes and more info on computomics.com. If you have questions or want to propose a next guest, please reach out to us at podcast@computomics.com.